Welcome everybody to the Beckman Directors Seminar for Thursday, November 5th. My name is Jeff Moore and I'm the director of the Beckman Institute. It's really my pleasure. I think you're all going to really enjoy today's speaker. The Director's Seminar speaker for today is going to be Marnie Boppard, and she'll be telling us about her research after I give just a brief introduction. So first of all, thanks for all of you to join us and hear about some of the activities and research that's gone on at Beckman through the Director's Seminar, which is really the main reason we have the Beckman Director's Seminar on a monthly basis. As our meetings have gone virtual, the format that we'll use is the Zoom chat feature. So please feel free to engage in that. And I know you can even have dialogue between yourselves, and it's been for my group actually a pretty interesting way to really facilitate and stimulate conversation and discussion. I know Marty will come back and look at the chat, or Patty will help us feed through it at the end. And I think it's a great opportunity to have some pin your questions and get them and try to get them answered as you think about them. Professor Marnie Boppard is a professor in Department of Kinesiology and Community Health. She heads the Molecular Muscle Physiology Lab here at Beckman, and she also leads a very exciting activity, the Extracellular Vesicle Imaging and Therapy Working Group. And it's really been a pleasure for me to see that come together, an area of emerging science and technology, and having a person like Marnie pull together a community and watch that unfold has been a true delight. And I look forward to what's going to come in the future, because I do believe it's an exciting and emerging area. Marnie's been a leader in many other ways. She's clearly an interdisciplinary scientist affiliated with both the Department of Cell and Developmental Biology. She's a affiliate of the Carl Woese Institute for Genomics Biology and is faculty on the Carl Illinois College of Medicine. Marnie earned her degrees in anatomy and physiology from Boston University, a postdoctoral study at Harvard Medical School, and also at the University of Illinois. And her current research focuses on understanding the molecular basis for muscle adaptation and exercise, which is why I think it's such a great talk, and while you all be able to relate to it, because I think we all are wanting to make the most out of the time we spend keeping ourselves physically active, and thinking about what is the molecular basis of that is just a super exciting topic that really, I think, cuts into the heart of how we all will be able to relate to the talk that Marnie's going to give today, Pursuing the Fountain of Youth, Development of Novel Therapies to Rejuvenate Aged Skeletal Muscle. So Martin, thanks so much for joining us. We're really excited to have you.
Awesome. Thank you very much, Jeff. I really appreciate that introduction. So before I get started, I wanted to thank a few members of the Beckman Institute, especially our Beckman directors, Jeff Moore, who you just heard from, and Patty Jones. Of course, thank you for the invitation to present today, but also for your guidance and unwavering support over the last several years. Really appreciate it. Dax Shearer and the animal facility for the great care that they provide for our mice, both young and old, and I'll tell you about those today. And then Daryl Spildman, who's a project manager in the Steve Boppard lab. Anytime we have a computer or equipment break, he's right there to fix it. And we're just so grateful to have him here at the Beckman Institute. And then finally, I'd like to thank our former and current administrative assistants, Julie McCartney, Kelly McKinley, and Bill Mullins. I really don't know what we would do without any of them. I have been coming to the Beckman Institute, and I've been working here. It's kind of crowded in the office at home. And I have to say that I really miss seeing these crowds here in the Beckman Institute in the atrium. Right now we have the testing center set up, but really missing these crowds, and I hope we can get back to some normal soon. Okay, so just to begin, Jeff already provided an introduction, but I thought I'd start with just some basic information about my background and my overall research goal. So as he mentioned, I am a professor in the College of Applied Health Sciences, Department of Kinesiology and Community Health. And our college has a long and interesting history in the areas of exercise, disability, and aging. And this is primarily due to the prior leadership of T.K. Curitin, who was a former faculty member, and he was considered the father of physical fitness, as well as the efforts of a pioneer, Tim Nugent, who transformed our campus into the most accessible in the nation. So very proud of my home college and department. As Jeff also mentioned, I'm currently an active member of 2 interdisciplinary centers on campus. One here in the top right is the Institute for Genomic Biology, but of course also the Beckman Institute. And so what do I enjoy most about the being here at the Beckman Institute? Well, it's these individuals that are pictured here in the bottom right. So these are some of my collaborators, my husband, Stephen Bopart, Vivash Debrucki from bioengineering, and Justin Rhodes in psychology and neuroscience. And what I really like about working with them is their high level of curiosity, their desire to explore and break boundaries. So I really appreciate just the opportunity to work alongside with them. So the primary goal of my research is to develop biological products that can promote healthy aging. And even though we work at the preclinical level with mouse models, we hope that the information that we generate can help people live long and productive lives. And there are 2 people in particular that have inspired my career. They're shown here. First, my mom, who's almost 80, in great condition, still a natural redhead, as you can see. But when I was growing up, she was a single mom, and she worked very hard as the director for food services in hospitals and mostly senior care centers. And so I used to accompany her to work, and it was there that it was my job to entertain the older adults in the dining room. And it was also here that I observed a wide variety of abilities and disabilities. So there were individuals, older adults, that were very active and engaged and excited. And unfortunately, though, there were others that were very quiet and in wheelchairs in the corner. And I think that this really impacted me in such a way that I've always been interested in understanding how we can maintain health across the lifespan. And so it was this experience then that allowed me to begin PhD studies under the mentorship of Roger Fielding, who's now at Tufts Medical Center. And he really is a pioneer in the area of understanding how exercise can impact health span in older adults. Okay, so my first grant as a PI here at the U of I was from the Ellison Medical Foundation, a New Scholar Award, and this was really quite an honor. And part of the requirement of this award was to attend a 1-week symposium every year in Woods Hole, Massachusetts. And it was there I was able to interact with experts in the area of aging from around the globe. And so by sitting through all these different lectures and talking with these individuals, I came to have a really good grasp of the biology of aging. So we understand that it really does begin with DNA damage. This is the first stage of aging. And it's due to just exposure to intrinsic and extrinsic stressors. So you can imagine like exposure to UV light can cause some DNA damage. And so here we have genomic instability occurring, telomere shortening, and epigenetic changes. Now, as a result of DNA damage, the cell will evoke a stress response that in the short term is helpful. So for example, we know that the mitochondria will start to produce reactive oxygen species, and the cell will undergo a state of cellular senescence or permanent state of cellular arrest, cell cycle arrest. And again, in the short term, some of these changes are very beneficial, but chronically, they can be very damaging to the cell. So as ROS starts to accumulate, it can cause destruction to the cellular structure, and the senescent program can allow these cells to start producing pro-inflammatory cytokines, allowing for a shift to this last stage of aging, which is a state of systemic inflammatory condition. Okay, so we know that there isn't a tissue in the body that is spared by the aging process. However, there are some tissues that are more sensitive than others. So in the aging field, we tend to focus on the eyes, the vessels, the brain, as well as my tissue of interest, skeletal muscle. And I think that the importance of skeletal muscle for health is really underappreciated. We know that muscle in a healthy individual makes up almost half of a person's body weight. It's the second largest organ system after the skin. And it is vital for a variety of essential functions. So we know that skeletal muscle, of course, is important for movement. So we can explore the environment and acquire resources such as food. We know that it's necessary for respiration, bringing oxygen into the body. We know that it's necessary for proper glucose disposal and metabolism, as well as maintenance of thermoregulation. And a small or a large amount of loss, it doesn't matter, can result first in metabolic disease, such as type 2 diabetes and cardiovascular disease. But then inevitably it can significantly increase our risk of disability. So we know now that approximately 40% of people aged 65 years or older report at least one disability. And you can see some of the different categories of disabilities that we can acquire with age. But I think that you can appreciate here in this chart that musculoskeletal conditions are the primary cause of disability in older adults. So here what we're talking about is the loss of skeletal muscle mass, loss of bone mass and mineral content, and also degeneration of our joints. And collectively, this can allow then for the progression of sarcopenia, weakness, fatigue, increased susceptibility to fractures and osteoporosis, and a debilitating condition known as arthritis. Now, if we just focus on muscle again, and if you look at the chart here, you can see that it's estimated that we lose about 1 to 2% of muscle mass every year after the age of 50. So we just follow this dotted line. You can see that this is the normal progression of muscle loss in the absence of significant injuries and disease. Now, we make really good choices about diet and exercise. We can significantly improve this progression and prevent disability, but as you can see, we can't fully mitigate it. There's always going to be some loss. Now, unfortunately, most individuals in the United States follow this lower trajectory of muscle loss. And so as we age, surgeries and illnesses become more likely and more prominent, resulting in limb immobilization, such as casting, short and long-term periods of bed rest. So we contract COVID-19, it's inevitable we might be laying in bed for a long period of time, and of course, losing significant amounts of lean muscle mass. Now, as you can appreciate with the chart here, with each one of these events, there's going to be an exacerbation of muscle loss and an acceleration towards the onset of disability at a relatively early age. So this gray area is the entry into disability. And you can see that the onset is between 60 and 70 years of age versus say 80 or 90. So clearly it's going to be important to recover any of the muscle that's lost during each one of these events so that we don't actually fall into that category of disability. So how can we recover the muscle? Well, there's no magic solution. There's no magic pill. The most effective approach that we know to recover muscle loss is engagement in resistance exercise. So you know that resistance exercise is the most effective approach to rehabilitate muscle after disuse. However, as you can imagine, there are significant limitations to the ability for older adults to engage in physical activity or recommended physical therapy. And the main reasons are problems and issues regarding mobility, and balance, and pain. So all of these issues can, decrease the comfortable, level with engagement in exercise, as well as tolerance for exercise. And then also what we know is that as we lose muscle mass, we actually decrease our capacity for growth. So as we lose our muscle, it becomes harder to regrow that muscle that's lost. And this is known as anabolic resistance. And this concept has been supported by many studies, both in rodents and in humans. But I think it's really best exemplified by this study that was conducted by Charlotte Saweda from the University of Copenhagen back in 2013. And so here, all she did was she recruited young and old individuals to the lab, and she subjected them to unilateral casting for 2 weeks. And so she just placed a cast on one of the legs for a total of 2 weeks. At the end of the 2 weeks, she took a muscle biopsy and evaluated the type 2 fiber cross-sectional area, the size of the individual fibers within the muscle. And you can see that there is a significant decrease in fiber size in both the young and the old. After that 2-week period, though, she had these individuals engage in strength training for 4 weeks. And as you can see here, whereas there was full recovery in the young, there was actually no recovery in the old. And so this is a really hot topic in the area of skeletal muscle biology and physiology. We want to know what is responsible for this lack of regrowth that has been observed. And then secondly, and very importantly, we want to know, can we develop therapies to boost regrowth, accelerate recovery, and ultimately prevent disability? Okay. So my approach to trying to address this gap in knowledge is to study the cellular molecular mechanisms that drive skeletal muscle adaptation in response to exercise, and then determine if deficits exist in these mechanisms with age. So our unique research agenda over the years has primarily focused on the alpha-7 beta-1 integrin complex within muscle. So if you're not familiar with the integrin, this is a transmembrane adhesion, a molecule. It's actually a heterodimer of an alpha and a beta subunit. And its primary function in muscle is to attach the extracellular matrix, specifically laminin, to the intracellular, to the interactin cytoskeleton within the fiber. And then using a genetic mouse model for the integrin, so integrin overexpression, alpha-7. So here we're going to overexpress the alpha-7 BX2 integrin subunit, specifically in muscle. We have found that this allows for the muscle to promote the process of muscle growth and vessel growth. And so we have published several studies over the years that demonstrate an important role for the integrin, serving as a mechanosensor at the muscle membrane, promoting muscle growth. We just submitted 2 papers demonstrating first that the integrin can promote muscle growth via a mechanism that does not include or is not dependent on the canonical pathway mTORC1. And secondly, we have found that, and we submitted a paper, actually resubmitted a paper, demonstrating that integrin localization at the membrane is disrupted with age. So we believe that there is an important role for the integrin in the process of anabolic resistance with age. Now, one observation that I made using this mouse model several years ago was that there's an accumulation of stem or stromal cells in the muscle based on this general stem cell marker, stem cell antigen 1, mainly around the vasculature. And so you can see that in this image of a muscle and cross-section with the red fluorescence. These are the cells that are accumulating around the vascular neurobundle. And so we knew at this point that these cells were not the primary myogenic stem cell for muscle, the satellite cell, because they did not express PAX7, but that these cells were either the fibroblasts, which are connective tissue cells that mainly produce collagen, or more likely that these cells were pericytes. Now, if you're not familiar with pericytes, these are vascular stromal cells, secretory cells, that surround the endothelial cells of our microvessels, including arterioles, capillaries, and venules. So if you look here in this illustration and in the center, specifically, if you look at the cells shown in green, we know that these pericytes are characterized by having a large cell body and sending multiple projections that wrap around the endothelial cells, actually forming tight connections with the endothelial cells in the form of adhesions and gap junctions. And because of the localization of these pericytes near the vessels, we know that they play an important role in many vascular processes, such as they regulate vasodilation, constriction processes. They can regulate permeability of substances across the membrane. as well as they can regulate an important process, angiogenesis. Okay, so based on this observation, in this animal model in which we saw that there was promotion of growth in response to exercise, we decided to outline three main goals that we wanted to accomplish. And the first was just to simply identify this perivascular stem or stromal cell in the muscle that appeared to be responsive to integrin overexpression and exercise. And then we also wanted to determine if the cell type could allow for the promotion of muscle growth in response to contraction. Once we identified the cell type, then we wanted to determine the extent to which the cell was dysfunctional with age and or disuse in the form of immobilization. And then finally, we wanted to evaluate the potential for cell replacement to recover the muscle following a period of disuse. Okay, so to address that first goal, we decided to develop an electrical stimulation-based model of resistance exercise that could be used with mice. So if you've ever worked with mice, the mouse model, you can imagine it's really hard to get them to lift weights. Okay, so you can't just simply put weights in their cage and have them lift. And so this was an alternative solution. We're going to, under anesthesia, we're going to place electrodes on either side of the sciatic nerve that is going to then excite all of the different muscles in the hind limb of the animal. And so these are 4-month-old adult C57 Black 6 animals. And once we stimulate the nerve at 100 hertz, because the foot is attached to a rotating pedal, the mice will complete 20 concentric and 20 eccentric contractions with full rotation during approximately 20-minute period of time. And so following this acute bout of exercise, we then collect all of the hind limbs of the muscle at 24 hours post contraction, and then we process the muscle for multiplex flow cytometry. So after elimination of debris and doublet cells, so that we're only working with the mononuclear cells within the muscle, and then also eliminating all contaminating cells or cells we weren't interested in evaluating, such as the CD31 positive endothelial cells or the CD45 positive immune cells, then we were able to evaluate excuse me, the relative quantities of pericytes based on 2 established pericyte cell surface markers, CD146 and NG2, as well as our PDGF receptor alpha positive fibroblasts. And so the data, the summary of the data is here on the right-hand side. The blue bars represent the relative quantities of pericytes and fibroblasts in the sham condition or non-contracted condition, and the red bars are the relative quantities after the exercise bout. And so what we found was that it was pretty clear that you can see here was that the CD146 positive pericyte quantity comprised the largest relative fraction within the muscle, anywhere from 50 to 70% of the total gated fraction. And this is in contrast to the NG2 positive pericytes in the fibroblasts, which comprise only about 5 to 15% of the relative fraction. In terms of then the quantity in response to electrical stimulation, we found that there was a small but non-significant rise in pericyte quantity in total. And this result was very consistent with some data that we published in humans following eccentric contraction. And this was in a publication back in 2015. Okay, so no major changes in quantity. However, in terms of function based on gene expression, we found that there were significant alterations with these 146 positive pericytes. So the next step was, in fact, to evaluate the cell function in response to contraction. Here, we're looking at 3 hours post-stimulation. And so first, we had to optimize the conditions for the isolation of these 3 different cell types using fluorescence activated cell sorting. And we were able to successfully complete that. And then we completed a high throughput quantitative PCR analysis, mainly focused on 70 genes. So this is a targeted analysis of 70 genes that we selected because they were involved in the processes of muscle growth, angiogenesis, ECM remodeling, differentiation, and neurogenesis. And I'm not showing you all of the data here that we collected. That data has already been published last year. But we found that the NG2 positive pericytes and the fibroblasts were minimally responsive to contraction. And this fibroblast response, this non-response, was really surprising to us. However, in contrast, what we found was that these CD146 positive pericytes, which are in high quantity in the muscle, were highly responsive to the contraction event. So we found that there was a significant increase in the gene expression of FGF2, as well as TIMP1, which suggests that these cells might have a role in initiating angiogenesis as well as arteriogenesis. We found that there was a cluster of genes that were upregulated related to ECM remodeling, including TIMPs and MMPs and collagen. And then finally, we found that there was a significant increase in the nerve growth factor, NGF, as well. And so based on these results, we decided to complete an unbiased evaluation of pericyte gene expression following contraction using RNA sequencing. And so of the 329 genes that were differentially regulated in response to contraction in these cells, we found that there were several genes that were significantly changed that were important for processes such as, again, angiogenesis, ECM remodeling, and to our surprise, also immune cell regulation. And so some of these genes included the interleukin receptor antagonists as well as pentraxin-3. And both of these genes we know are important in mediating an anti-inflammatory response. In addition, we found that there was upregulation of HMOX1, which encodes for heme oxygenase 1, suggesting that perhaps these cells might be mitigating an antioxidant response. So based on these gene expression profiles, it appeared that the pericytes in the muscle were working very similarly to mesenchymal stem cells, which have been characterized in many other tissue types, such as the bone marrow, skeletal muscle, and adipose tissue. Specifically, releasing factors such as anti-fibrotic, pro-angiogenic, and anti-inflammatory factors that can facilitate repair and remodeling and regrowth of tissues in response to injury. Okay, so we completed then one last experiment in support of this first goal. We wanted to determine the extent to which these pericytes could enhance or initiate the process of skeletal muscle adaptation in response to contraction. And so here, again, we took our wild type animals, adult animals, and we divided them into three primary groups. First, a total control that received no treatment. A second group that received bilateral injection of PBS or saline serving as a second control. And then a third group that received bilateral injection of the CD146 positive pericytes. These second two groups then were subjected to electrical stimulation-based training 2 times per week for a total of 4 weeks. And what we found then, if you look at the red bars, these are the results for the animals that received the pericyte injection. You can see that there was a positive shift in the myofiber size such that there was a decrease in the small size fibers in the muscle and enhancement of the larger size fibers within the muscle. In addition, we found that with the enrichment of the pericyte in the muscle prior to the training bout, we found that there was a significant increase in ECM remodeling based on a CHP assay, which can measure degraded collagen within the muscle. So it seemed like the matrix surrounding these fibers was turning over at a greater rate. And then finally, very much in line with a role for the pericyte in that process of angiogenesis or new vessel formation, we found that the pericyte enrichment facilitated an increase in capillary density. Okay, so based on these results then, we felt that it was the pericyte that was highly responsive to exercise. And our experiment suggested that it was this cell type that could contribute to skeletal muscle adaptation in response to contraction. So this led us to that second goal, which was to determine the extent to which this periocyte was dysfunctional with age and or immobilization. Okay, so our approach here was a little bit different. We decided to complete a comprehensive analysis of all of the different cells, mononuclear cells within the muscle, using the single cell RNA sequencing approach. So if you're not familiar with the 10x Genomics platform, it begins by dissection of your tissue of interest. For us, it's the TA muscle that was collected from adult 4-month-old animals and aged animals 24-month-old. And so then after we dissected the tissue, we could dissociate all of the different cells, the mononuclear cells within the muscle, using a protocol that we developed that could help separate the endothelial cells from the pericytes. We then worked with the Biotech Center to complete the cell partitioning and cDNA barcoding, as well as library construction and sequencing. And in the end, we were able to perform cell clustering analysis using several established markers. And based on that, we could create this heat map, which identifies 14 different unique cell cohorts. And so here, for example, this cluster number 4 allows us to identify these cells as pericytes. And then here for cluster number 5, we were able to identify our fibroblasts. Okay, so we look at these individual cells in 2D then using our t-SNE plots. First, I wanted to point out that there was a decrease in the quantity of the primary muscle stem cell, the satellite cell. So you can see compared to the young adults, there was a decrease in this particular cluster. And this has been previously reported. And so this is just nice verification that this procedure worked. Interestingly, we did find that there was a significant decrease in pericyte quantity in the aged muscle as well compared to the young adults. So now if we just look very closely at the pericyte cluster and we examine gene expression, comparing the young adult versus the old, what we found for our top responders was an upregulation of genes responsible for the major histocompatibility complex, or MHC proteins. So this would include our H2Q7, Q6, K1, as well as something known as the beta-2 microglobulin, or B2M. And what's really interesting about B2M is it's an established pro-aging protein. So that we know that it accumulates in the blood of both rodents and humans with age. And if you inject it into a young mouse, it can cause neurodegeneration in the brain. Now, interestingly, in the literature, it has been suggested that if you genetically overexpress these MHC proteins, this can elicit an increase in p53 activity, which would promote cellular senescence and apoptosis. So that might explain why some of these cells are lost with age. And if we look at our gene list too, we find several other genes that suggest, again, that these cells are undergoing the process of cellular senescence. This profile was actually unique to the pericytes, as well as one of the different clusters of the endothelial cells, suggesting that these muscle-resident vascular cells are highly prone to cellular senescence and apoptosis with age. Okay. So we wanted to also take the same approach of evaluating all the different mononuclear cells in the muscle using the single cell sequencing analysis following immobilization. And in our laboratory, we have a model of immobilization that we use with mice. It's called the staple-based method of hind limb immobilization. And so here, we simply just tether the ventral part of the foot to the shin in full dorsiflexion, causing shortening of the tibialis anterior muscle. And this elicits a significant atrophy, decreased fiber size. We complete this procedure for 14 days. So only one of the limbs is going to be tethered, and they can still really make their way around the cage just fine. It's not a problem for the mice. And then at this 14-day time period, we will remove the staple and they'll start to put some weight on the limb, which we refer then to as the remobilization period. And they can do this for an additional 2 weeks. And what we found was that there was still significant deficits in terms of the fiber size within the muscle. So there was really very little improvement in the muscle atrophy condition, even after 2 weeks of recovery. So for this experiment, though, we removed the muscles from the animal, both the mobile limb and the immobile limb, at 14 days and completed the sequencing. And so in terms of quantity, we found there were no major changes in cell quantity. For the pericytes, it looks like maybe there was even a slight increase, but it's hard to say. So no major change in quantity. But when we evaluated gene expression in all of the different cell populations, so not specific to the pericyte, but all of the mononuclear cells, there was a decrease in gene expression of mitochondrial genes, so specifically genes responsible for mitochondrial structure and function. So for example, we found that there was a significant decrease in the mitochondrial rRNA expression. There was a decrease in the 2 of the different subunits for complex 1, as well as a decrease in the subunit responsible for or involved in ATP synthase. And so this would suggest that there is mitochondrial dysfunction within the pericyte, but in all the other cell types. And this really makes sense to us because if we're not contracting the muscle and there's no need for energy, these mitochondria are probably undergoing turnover. In addition, and much more specific for just the pericyte, we found that there's a decrease in contractile proteins, including the tropomyosin, smooth muscle actin, and the myosin light chain. And this seems to be consistent with reports suggesting that the pericytes lose their contractility in the disease process, such as diabetes, such that, again, the pericytes detach from the vessels. So overall, for the pericyte, what we found was that the pericytes are losing mitochondrial function and contractility in response to immobilization. Okay, so taken together then, our results suggest that with age, there's a decrease in pericyte quantity, which aligns very well with the loss of the vasculature as well, that we have observed with age as well. And we don't know if it's the detachment or the loss of the pericyte that causes the vessel destruction, or if it's the loss of the vessels that can result in the loss of the pericyte. We don't have an answer to that question. But in addition, we found that both age and immobilization can decrease the function of these cells such that they start to become senescent and undergo an apoptotic program, as well as lose mitochondrial capacity and function. Okay. So these studies then provided the justification for evaluating then their ability to serve as a mechanism for recovery of the muscle following a period of disuse. So we wanted to determine if transplantation of these cells following disuse might have any capacity on the recovery process. And so this work was completed by a former PhD student, Michael Monroe, who actually optimized this immobilization model for the lab. And so what he did here was then he placed pericytes from healthy animals into these recipient animals following 14 days of immobilization. He then assessed the muscle 28 days later to see if there's any improvement or recovery of the muscle at this time. And I want to be sure that you understand that this placement of the pericytes at this time point was really intentional. We hypothesized that because the pericytes are mechanically sensitive, so they're sensitive to that process of contraction, that during remobilization, these pericytes might start to synthesize and secrete beneficial factors that would help with this recovery process. Okay, so that's exactly what he found. So here in the blue bars, this is showing the data for the animals that received just a control vehicle. And you can see that there's still deficits in the myofiber CSA at this 28-day time period. However, when he injected the pericyte in the muscle, he found that there was full recovery of the fibrous CSA. And in addition, he found that not only was there recovery of the capillary density, but there was also enhancement of capillary content within the muscle as well. So we're really excited about these results, and we published the data in the FACET journal just last year. Okay, so Yu-Fu Wu is a current graduate student in the lab who has a specific interest in developing these therapies that specifically can help regrow muscle in aged animals. And so in our last experiment, we were using our adult animals, but here he wanted to determine if the pericyte could enhance recovery in these older animals. And we were both we had some reservations about these experiments, mainly because our lab has utilized a variety of stem and stromal cells. And we have found when we inject them into aged mice and they encounter that really hostile microenvironment, collagen enriched and filled with inflammatory cytokines, that these cells can lose their viability and they lose their function, and they're also prone to rejection by the immune system. And so that's exactly what happened. When we injected these pericytes into the muscle of these aged animals, we found that there was absolutely no recovery of the myofibrous CSA or capillary density. So is this at this point that I decided, okay, we need to take a different approach, maybe think about more creative means of delivering some type of therapy for the aged muscle. And so I started to read papers that really talked and focused on extracellular vesicles, or EVs. And if you're not familiar with EVs, these are nano-sized membrane-bound vesicles that are released by nearly all cells. So cells will release EVs in response to physiological, healthy stresses such as exercise. The cells will also release EVs in response to pathological stress, such as disease, cancer, Alzheimer's disease, heart disease. And these EVs can be produced by two different biological pathways, resulting in the production of a larger EV, which is known as the microvesicle, or a smaller EV known as an exosome. But it doesn't matter, because both of these types of EVs can transfer intracellular materials such as proteins and peptides, lipids, and nucleic acids such as miRNAs from donating cells, which are shown here, to either nearby cells or distal cells. So we know that EVs that are released by cells can enter the circulatory system and impact cells distal to the tissue. Now, as I was reading some of the literature, I came across some papers that demonstrated that healthy mesenchymal stem cells, which again are phenotypically very similar to the pericytes that we study, can secrete EVs that can reduce reactive oxygen species and inflammation, two of the hallmarks of aging. And so, of course, wanted to ask the question, well, do the pericytes secrete EVs? And I can tell you, and I won't show you the data, but yes, in culture, we know that the pericytes do secrete EVs in abundance. But we also wanted to know, do they elicit the same benefits, such as these MSCs, in terms of reducing reactive oxygen species and inflammation? And we know that the EVs are very stable within tissues. And so we believe that perhaps these would not have those same viability issues as injection of the pericytes. And so we wanted to also start to ask the question, well, if they could elicit some of those benefits, how can we isolate these therapeutic EVs, mainly from a biological source? Okay, so how could we specifically isolate the pericyte-derived EVs from blood so that we could collect them in a large amount, a significant amount, enough for therapy? And so this is really quite a challenge. Trying to identify subfractions of EVs is one of the major challenges in the EV biology field. Okay, so this was our solution to isolating the pericyte-derived EVs from the serum of animals. And so TBX18, just a few years ago, was identified as a unique transcription factor that is expressed by pericytes. And so we acquired the TBX18 cray mouse and we crossed it with a fluorescent reporter, a Rosa 26AA9 mouse, so that when we inject this double transgenic mouse with tamoxifen, we can conditionally evaluate the fluorescent pericytes inside the muscle. And so you can see in this cross-section of muscle tissue following the tamoxifen injection, we can start to visualize these TBX positive pericytes surrounding the fibers. So this is really cool because we can now use this mouse model to perform lineage tracing. So we can determine the fate of these pericytes following the conditions of exercise and disuse. But we didn't want to limit ourselves there. We thought, okay, well, this is really great. From these animals, then we should be able to isolate TD tomato positive EVs from the blood. And so that's exactly what we're doing right now in collaboration with the University of Ottawa. We are using these animals and collecting these TD tomato EVs using a process known as NanoFacts. And once we acquire these EVs, we can inject them into our aged animals. Okay, so while we're waiting for those experiments to be completed, we decided to work on one experiment, which has really turned out to be quite interesting. And so here we decided to isolate EVs from a healthy adult donor mouse. So these are EVs that were isolated from the serum of just normal wild type animals. And then we also collected EVs following the exposure of these wild type animals to the acute bout of exercise that I previously described. So we refer to these EVs as the EXERVs. And so we injected both of these types of EVs into the mice following immobilization and then analyzed the muscle at day 28. And what we found was that whereas the sedentary EVs had a minimal effect on the fiber CSA, the EXERVs not only restored the growth, the muscle growth or CSA, but actually enhanced growth to some extent, suggesting that these EXERVs are therapeutic. And so this has really captured our attention because we think that, you know, if these EXERVs are therapeutic, maybe this is our solution towards developing a therapy for the aged animals. And so we have just started to probe these EXERVs to determine how their profile is really different. And so we can, you know, take a proteomic approach, we can complete deep RNA sequencing. But even before that, we decided to work with the NanoView company using the ExoView R100, which allows us to acquire information about individual EVs within our samples. So we could look at the size, we could look at the content, we could look at cell marker expression. And what we found was that if you look at these images here, these are the results showing the EVs from the sedentary serum versus the exercise serum. And here on the top, we're looking at EVs, and this is on a microchip, that express CD9, okay, or CD81. These are common EV markers. And what we found was that when we collect the serum 90 minutes following exercise, there was a significant drop in CD9 expression on the EVs and a 5-fold increase in the expression of CD81 on the EVs that we had collected. And so what's really interesting about CD81, even though that it is just a typical marker for EVs, we found in our single cell sequencing results that the pericytes and the satellite cells specifically express CD81 to a large extent. So there is this opportunity for us to then determine if our pericytes secrete EVs that express CD81, and perhaps this is our best approach to identifying our therapeutic EV. So now we can start to think about too, if these CD81 EVs do represent pericyte-derived EVs that circulate in the bloodstream following exercise. We can ask what impact do these EVs have, not just on the muscle, but on other tissue types, such as the brain. And that is the basis of my collaboration with Justin Rhodes. Okay, so just to conclude then, what we have learned so far in this particular area that I focused on today is that the pericytes within the muscle are highly responsive to contraction and positively initiate skeletal muscle adaptation. And most importantly, we believe that these pericyte-derived EVs, perhaps also the XRVs, may represent an effective strategy to recover muscle mass after disuse in older adults. So here's my last slide. I just wanted to mention that, going back a few years ago when I started to become interested in EVs, I started talking with other faculty, including my husband, and it became clear to me that there were a number of individuals also interested in extracellular vesicles. And we had the same questions. How do we isolate? How do we characterize? How do we track these EVs? And so that was the justification for developing the EVIT working group, which is still ongoing, very active. We have group meetings every month, and we hope to have another workshop in the spring. And with the support of Jeff Moore and Patty Jones and the Beckman Center, we're developing an EV resource center. So a hands-on lab where students, investigators can come and learn some of the best practices in the area of EVs. And so as a result of this working group, we've established, of course, many collaborations, which you can see here. But even better than that is bringing other faculty together. I noticed there are teams that are building and really, synergizing and really sharing information. And I think that's what Beckman is all about, really initiating collaborations that help us to reach our goals, just individually as well as across campus. So none of this work would have been possible without my students. I just wanted to really thank Svatoslav Dvoretzky, who's shown here. He graduated in May. He now works for Abbott Nutrition, and he initiated all this EV work in the lab. And then Yufu Wu, who again is a current student, now continuing this particular research. I'd like to thank my collaborators, especially those at the Biotech Center, and then of course the funding that allowed this work to be possible. So thank you, and I'd be happy to take any questions that you have.
Thank you so much, Marty. On behalf of Jeff, who had to leave early to go to one of his own students' PhD defenses, he wanted me to express to you his great appreciation and enjoyment of this excellent seminar. So thanks again for, I think, striking just the right balance of technical information and a broader perspective for a very diverse audience who can appreciate the quality and relevance of your work. So again, thank you very much for doing this for us. So audience members, I'm going to go over here and, let you guys unmute yourselves if you would like to ask questions verbally, or you could always put questions in the chat, and Marty and I can both see the chat, or if you want to just privately chat to me, Patricia Jones, on the chat, and I can ask the question for you. We're happy to hear whatever your questions and comments are. One of my basic questions for you, Marty, just to kind of kick this off, is what sort of studies have been done in humans that are somewhat analogous to this? So there's always this question of, how well does an animal model predict what happens in people? And so can you comment a bit on what the literature or other studies have shown related to humans?
Yes, there are only a couple that I'm aware of where they have looked at the proteome of the EVs. And they have found that there are distinct changes that occur in the EVs when they're collected from the serum of humans post-exercise. And what's interesting is they don't show any overall quantity changes. It just seems to be like in their hands there was just this change in EV quantity. And I don't know if there's so much that we don't understand about EVs so far, and the response to exercise. Are some EVs absorbed really quickly by tissues while others are secreting, and that's why we don't see any quantity changes? It's really hard to say, but But again, they did complete a proteomic analysis, and they did find that there's significant changes occurring in the EVs. Other than that, we really haven't seen a whole lot of human studies that completed so far, which is surprising because I think that, I've always been an animal researcher, preclinical, researcher, but it's at this point where we're seeing some very striking findings, and we're ready to pursue some of the same questions in humans.
Thank you. Oh, yes, here's a question sent to me from an audience member. Is there a difference in EVs from aerobic type versus more intense muscle exercise?
Great question. Yeah, we don't know because we haven't addressed that yet, but that's very much in line with some of the questions that we have as well. So my guess would be that yes, there are probably differences in terms of EV content and capacity for adaptation, but that has yet to be explored. A brand new area.
Well, that's always great to get ideas for new future grants and
yeah.
things. Could you tell us a little bit more about the lab you're setting up at Beckman? Like what equipment is in there, and to what extent does commercial equipment help you do your research versus what kind of instruments should we be imagining to build for the future?
So do you specifically mean the EV Resource Center?
Yes, EV Resource
Center. So we do have the nano tracking analysis here at the Beckman Institute, which allows for, a quantity evaluation of the EVs. However, there are some limitations with NTA now that we're starting to understand. It's sort of the NTA analysis will select the larger EVs for quantitation. It discards, some of the smaller EVs. And so we'd like to acquire a Spectrodyne system. So it's based on a very different technology for evaluation of the EVs, and doesn't have that same limitation. We're also, we've been in touch with the ExoView company, the NanoView company, and we are, based on the results that we've obtained, we'd like to acquire that system as well for characterization of the EVs. And then, past that, what we hope to do is to be able to, provide that hands-on training that allows investigators to have a good sense of how do you use either ultracentrifugation or now it's becoming more popular, the size exclusion chromatography method for isolation. And past that, just flow cytometry, ELISA-based methods, and Western blotting for characterization, verification that you have in fact collected your EVs. So we're hoping to include, all of that, plus have a director. So we're writing a center grant right now to form this research center, so it can be staffed with individuals, not just individuals from my lab, but a real expert in the area of EVs.
Oh, that's great. All right, thank you. Let's see, we've got 2 minutes left, so let me just see if there's any 1 or 2 more questions from the audience. So feel free to speak up, unmute yourself, or send me a question in the chat, or just make a public chat so we can all see the question. I'll just wait a few seconds here. Okay, I don't see anything else. So I think, again, thank you very much, Marty, and thank you all for being with us today. Please join us again for our next Beckman seminar. And please keep checking our website where we will post news and other things related to all the research going on at Beckman, including Professor Marty Bopart's work. So again, thank you very much, Marty, and thanks to everybody for being here.
Thank you so much, Patty. Appreciate it.
Thank you.
For the opportunity.
Thank you. Bye-bye all.